All right, let's go ahead and get started. Thank you everyone for joining us. I am here with Charlie Yoakum. Today's sound learning class is COVID-19 vaccines, how they are made and how they work. Before I hand it off to Charlie to let him introduce himself and give a little bit more background, I just have a few ground rules. A lot of you are familiar with these already. There is a raise hand button at the bottom if you have any questions. I'll be the one keeping an eye out on those. And if you have a question throughout the presentation, click the raise hand button and I'll allow you to speak and ask the question. Another ground rule is towards the end of the webinar, we will open it up for a poll. So if you could please stick around for that for me and just do a survey on this class. But other than that, I'm going to hand it off to you.
Okay. Thanks, Jordan, for keeping things in order here with our switches between Zoom and in person. As you can see from the title, we'll talk about COVID-19 vaccines, how they're prepared and how they work. Looking at the crowd, there's one. Okay, so we don't have coordinators, so we introduce ourselves. I'm a retired professor from the University of Michigan. I spent 37 years there teaching biochemistry, molecular biology, and chemistry to graduate students and to undergraduates. I trained 16 graduate students in their PhD work, and I trained 5 postdoctoral fellows who are now mostly in European positions in universities. My research was a little esoteric in that I was interested in trying to discover how photosynthetic reactions convert water into oxygen. That seems to be a little esoteric, but remember that everything you're breathing right now comes from water that's been processed by plants and turned into oxygen. And pelican sound, been seasonal here since 2009. And one of my neighbors is in the audience. Okay, so here's an outline. I want to talk a little bit about your immune system, the little bit that I know and a little bit that I could put together. I'm not an immunologist by any stretch of the imagination. I want to talk about the brief history of vaccination from smallpox to polio, because there's a lot of interesting history here and it sort of sets the stage for what we're going to talk about when we get to the COVID vaccine. I'll talk about Edward Jenner, who's the pioneer in this field, about John Enders and Jonas Salk and polio and measles vaccines, and what turned out to be the long experimental road to putting these particular vaccines together. And then the vaccine race, that is the COVID-19 base and race to get to the immunity that we have today. And it was basically a very safe shortcut. There'll be conclusions at the end and plenty of opportunity to ask questions if you have them. So let me talk about your immune system a little bit and give you a 2-part definition. There's an innate part of your system, which is the first line of defense. You've got some killer cells that are called macrophages that are kind of cruising around in your system looking for things to knock off that might harm you. Your hair, your skin, anything that can prevent the bad guys from getting in. That's the innate system. It's a rapid line of defense, and it's there to look out for you in the first case. The second part is the adaptive case. White blood cells, leukocyte cells, are the cells that detect antigens, synthesize antibodies, and neutralize them. Antigens are proteins, carbohydrates, various chemicals that can come in by way of viruses, by way of bacteria, insect bites, all sorts of things. Carbohydrates could come in as well on bacterial cells. These are all energetic compounds. They're not good for you. They can be external, like the wrong ABO blood group. If you get the wrong blood transfusion, say you're a type B and you get type A, you can provoke an immune response in your system that is not good for you. Viruses can bring it in, bacteria can come in. Another external effect, if you've heard of anaphylactic shock, that's an extreme reaction of your immune system to something like, let's say, peanuts or a bee sting or something of that sort. And that's something where you need to get help immediately. You got to call 911. You've got to get to an emergency room, and you have to get a shot of epinephrine or adrenaline, as it's known more commonly. Internally, rheumatoid arthritis is something where your own immune system is acting up against you. More, again, exoterically, transplanted organs. There's a lot of work that goes in, you've probably heard this, about matching a donor and a recipient for organ transplantation. Because the transplanted organ itself, once implanted, can be viewed by your immune system as a foreign visitor, and you may wind up getting rejection that's caused by immune reactions from your system. So this is very important, and that's why they use a number of drugs to do immune suppression in organ recipients. And these are individuals that are said to have these compromised immune systems that you've heard about on television when they're talking about COVID and the damage that COVID can do. So antibodies. Antibodies are produced by your immune system. They're proteins, and they have a very specific binding site that allows them to attack and neutralize an antigen. So once an antibody finds an antigen that it can interact with and it binds to that antigen, the antigen is essentially taken out of the system and it's neutralized. So we can talk actually about neutralizing antibodies, and that's probably popped up on TV, and you might have heard about that in the context of COVID-19 vaccination. So a diagram of an antigen-antibody interaction goes a little bit like this. The antibody is made up of two different proteins, this big long thing that's stretched out there like a Y. That's the heavy chain and a light chain, which is the little short thing that's attached to it on two sides. And the combination of that short light chain and part of the heavy chain makes up a recognition site that can look for antigens and can neutralize them. And that's what's shown in this diagram here. The antigens, again, can be a number of things that I've talked about: carbohydrates, proteins, various chemicals. And once that binding occurs between the antigen and the antibody, you've inactivated the antigen, and the antibody has done what it's supposed to do. Just to add one more thing to the pot here, the antibodies that are circulating in your system can be polyclonal or they can be monoclonal. And both of those are shown here. On your left is a polyclonal antibody with several binding sites, or polyclonal antibodies with ability to bind at several different sites. And this would be in your antiserum if you were to be bled, and we could get rid of the red blood cells and everything and have some serum. It would have these antibodies in it. And if you look at this carefully, you see that the two kinds of antibodies here. Some of those antibodies bind to the little pyramids, and some bind to the blocks or the squares. So the artist who did this viewed the antigen as a sort of looks like a potato, and these particular pieces of the antigen that are sticking out are called epitopes. You don't have to remember that, but there's an epitope antibody binding interaction. And you can see that here for the polyclonals, that you have two different antibodies that are attacking two different parts of the antigen. There are also monoclonal antibodies, and you may have heard of this. I'm sure you've heard of this probably in the news and everything. Monoclonal antibodies can be directed precisely at one binding site on an antigen. They can be prepared, in laboratories. They are commercially available. Regeneron makes a monoclonal antibody that was effective against the Delta variant of COVID. And they are specific for just one binding site. So you see this particular monoclonal antibody will only bind to the little pyramids on the antigen. So those are monoclonals. As we develop antibodies, we generally develop polyclonals. You have to do a lot of trickery with genetics and mice and so forth and so on to get to monoclonal antibodies that have their own uses in various kinds of therapy, cancer therapy, and in the case of the Delta variant of COVID, in treating, using it as a therapy, giving it to a patient as a partial relief from the virus. Okay, so the other part of your system, we've got to talk about three important cell types in your immune system. I don't have to dwell on this very long. There are B cells. These are the good guys that are capable of rapid antibody production. There are memory B cells. They kind of remember if they've seen, if their antibodies have seen that antigen before, and they're sort of ready to jump into the fray as quick as they can. And sometimes this, when I say quick, this is a matter of many days usually to develop a full complement of antibodies against a particular antigen from the B cells. But once you've done it, there's a memory in the system that says, you know, I've seen that. If it comes around again, I'm ready for it. You also have killer T cells. I like that name. Killer T cells are very unusual metabolism. They're able to recognize and destroy your cells that are infected by viruses. And they use some pretty fancy chemistry to do it. They release hydrogen peroxide. They release various killer chemicals, and they're a line of defense. And then they're the helper T cells. The helpers are just the good guys that sit around and kind of coordinate and assist in the production of the B and the T cells. Okay, so to do a quick summary of this, remember that you have an innate system that's really fast, your skin, your hair, macrophages in your circulatory system, and you have slower adaptive responses. And the adaptive responses are the ones that involve antibody production against antigens. And just to remind you, antigens, proteins, carbohydrates, transplanted organs, wrong blood groups, allergens, things like pollen, for example. So this system is extraordinarily flexible, and it stands ready to guard you against a number of things. Sometimes it does too good a job. Remember anaphylactic shock. But by and large, this is something wonderful that we all have, and it's something that looks after us. So the adaptive system is basically the system that is the target for vaccine preparations. Vaccine production takes advantage of and makes use of the adaptive system and anti-production by that system. Excuse me, never happened before. So the rest of today's presentation will be about the adaptive system. And the adaptive system is going to be at the back of everything we're going to talk about. So the next thing to do then is to go into the history of vaccination. There's a lot of interesting stuff here that I didn't know about actually when I was looking into this. Smallpox was a focus of inoculation because smallpox epidemics were quite common in the old days. Going way back into the 1600s and the 1700s, there was inoculation against smallpox. It was called variolation. And it was common in Asia and Africa and Turkey, and in particular in England. It caught hold in England and was very popular in England. And the name comes after what smallpox was called. Smallpox was called variola, okay? So what's variolation? Basically, people took the remnants of scabs, other pieces of the remains of a smallpox infection from victims. So ordinarily it was ground up into powder of some kind and then was used for inoculation, but not like we think of it today. One way of doing it was to take the powder and just snuff it up your nose. Another was to make a small cut in your skin and rub it in. The idea was that what you wanted to do was to induce a mild case of smallpox that would be sufficient to inoculate you against the really bad effects of the disease. And medical historians have looked into this in some degree, and it actually worked. From what I could find out, in a smallpox epidemic, 1% to 2% of variolated people would die, but 25% of unvariolated population would die in the same epidemic. So you're getting a pretty good defense here against smallpox using this rather crude method of inoculation. And excuse me, unbeknownst to me, in 1777, George Washington had his continental troops variolated so that they could stand up and match the British troops who had a high resistance to smallpox because of the popularity of variolation in England, okay? So, in fact, this is before the development of any sophisticated sort of vaccination. This variolation was quite popular, and smallpox was the target of it. So let's carry on with smallpox for a little bit, because again, it's in the history of vaccination generally. Edward Jenner is the hero here. He lived from 1741 to 1823. He was a British physician, and he's generally credited with development of vaccination that was directed at cowpox or smallpox. You got to mention cowpox along with smallpox, because this is how Jenner got into the vaccination game. All the pictures you see of Edward Jenner, they're kind of romanticized. This is Edward Jenner, and he's vaccinating a small child. This child happens to be the son of the Jenner family gardener. And he was the first person vaccinated ever anywhere. And I can assure you that in more modern times, you are not allowed to go out and just grab the first kid off the street to test a vaccine. That's not done anymore. We have lots of rules and regulation. And briefly, we'll show you the vaccination was to protect against smallpox. We'll talk again about cowpox, but smallpox was a really horrible disease. And you can see just in this case, that's a leg of a patient. The welts and everything that come from the viral infection. So what did Edward Jenner do? He was, to me, you read about him, and he was one of the first real physician scientists in terms of his ability to observe, draw conclusions, and to go for a therapy. In England, there were young women, milkmaids they were called, who worked with cattle. They milked the cattle, they were around the cattle quite a bit. And what Jenner observed was that these young women who developed cowpox sores that contained pus, that is dead cells and dead virus and DNA and so forth, you know that now, but didn't then. He noticed that they didn't develop smallpox. And this was quite something. In fact, this had a lot to do with the popularity in the end of vaccination against smallpox, because it had this ability to prevent you from being disfigured by the disease. So it was in 1796 when you saw the picture. Jenner took some fluid from a cowpox blister in a cut on the arm of a young boy, and let that go for a while, and then came back around with his gardener's son, and later showed him to be protected against both cowpox and smallpox by intentionally infecting him with smallpox. Okay. So smallpox vaccination spread like wildfire. I mean, this Jenner's approach, the way to do it, and everything else is the first real substantial, I think, anti-pandemic kind of treatment that had a global impact. In the United States, it got over here really fast. And by 1850, there were vaccination laws in Massachusetts. In other words, you got vaccinated against smallpox, or there are things you couldn't do, like go to school and so forth and so on. I think that the vaccination itself advanced beyond using fluid from cowpox sores, because by the time they got around to worldwide vaccination, they were using an attenuated virus in the inoculation. I was inoculated when I was a kid. Many of you probably were as well. And in the 20th century, it was widespread. And basically, in 1980, the world was declared smallpox free. I think vaccinations for smallpox ended about 1970, 1975, something like that. And there are said to be 2 repositories of smallpox virus left in the whole world today, which are just there sort of on standby. Okay, so in this case, if we look at smallpox, this is really the long drawn out way to develop a vaccine, because you go back to the 1700s, 1600s, all the way up, say, to the 1900s, the 1950s, 1960s. We're dealing with 250 years or so to get to where vaccination has taken hold and has really cleared the world of a particular disease. So that's a long time. Let's look at the shorter time, but it's still a long road, and to see what people went through in the 1900s and up into the 20th century. to develop safe vaccines. The driving force for this is something that you probably all remember. In the 1900s through the 1950s, there were polio epidemics that I remember very vividly that would kill 500,000 people or so worldwide when they occurred. And they left victims crippled because the polio virus, among other things, attacks the spinal cord. And Franklin Delano Roosevelt, the president of the United States in the 1940s, was a prominent example of this. For measles, there were infections routinely that got to 3 to 4 million people every year in the United States. I don't remember getting a measles vaccination, but I remember going to a measles party that my mother sent me to, and I had it and haven't had it since. So the presence of these diseases, the recurring presence of these diseases in our population here in the United States did create a demand for vaccines. And in fact, the March of Dimes was something that Franklin Delano Roosevelt was behind. A guy named Basil O'Connor started it to provide funding to support research on polio. And later in the 1950s, other scientists attacked measles, and we'll talk about that just a little bit in a second. So who are the people that had the primary roles in this long road to getting us to safe vaccines? There are a small number of people that you can signify as having very important roles. So on your left, these are all scientists who led the way to vaccines. On your left is a group of 3 people, John Enders, Thomas Weller, and Frederick Robbins, who all worked at Children's Hospital in Boston and shared the Nobel Prize in Medicine and Physiology in 1954, because they learned how to culture the polio vaccine and to obtain amounts of the vaccine that were sufficient to allow scientists to do experiments with the vaccine. And that's why they got the Nobel Prize. Outstanding work. The right is someone everybody should know. That's Jonas Salk. He was at the University of Pittsburgh, and we remember him for developing the first polio vaccine. Okay, so what had to be done, let's say between about 1949, 1950, that's when Enders and his group actually did their work getting the virus isolated and learning how to culture it and everything else, at the time when the polio vaccine first rolled out. So the first thing that anybody had to do that was going to get into this business was to learn how to isolate and grow the virus. So that, I said, was about 1949 in the case of the polio virus. And that was John Enders and his associates in the Children's Hospital in Boston. That's hard research. You have to put a lot of work into this. It's not easy. You have to kind of feel your way and understand what's going on. And you have to be very careful because you're growing a very infective virus. The second thing that has to be done is you have to treat the virus in one way or another to inactivate its pathogenic property. In other words, you have to find a way to get the virus back in a corner where you can kill off its ability to infect and make people sick, but where it can retain other properties that you can use to make a vaccine. In the case of Jonas Salk, he used a simple chemical called formaldehyde to treat the virus. When you treat a virus of any protein with formaldehyde, you really attenuate its properties. You do what's called denaturation, so that the polio virus treated with formaldehyde should be inactivated as a pathogen, but maybe it's now useful in making antibodies. So what you have to do then is, if your chemical treatment is really doing what you think it is, you have to test the modified virus to make sure that it's safe, and you have to do this first in animals. And in those days, it was generally done in rhesus monkeys. They were tested. If the virus didn't kill the monkeys didn't develop any of the symptoms of polio. You knew that the virus was safe, but that's okay for monkeys. You had to test it in humans, and this was also done. You have to prepare a vaccine using the modified virus, test it in humans, and ask, is it safe? And is it a strong enough antigen? And that was done in a larger population of children, basically. And Enders later went through this with the virus for measles. He inactivated it by a more sophisticated process of passage through various culture mediums. and weakened the virus to the point where it wasn't infective, but it turned out to still be an antigen. So then what happens is after this, so you get a nationwide vaccination procedure that basically has put polio out of business. Unfortunately, there still seems to be some vaccine around or some measles around, owing to people don't believe in vaccination for measles, for MMR. And that remains a bit of a problem. I remember an announcement last summer that someone with measles had passed through LAX in Los Angeles, and to be careful if you've been in that particular airport because it's an extremely infected virus. Okay, so anyway, from start to finish, meaning you get a good vaccine and vaccination begins, how long did this take? Okay, Salk got a real jump on the whole problem because of Ender's work in Boston. So he had the ability to culture the virus, knew how to grow it, knew what to do with it in such a way that he would have plenty of virus to work with. He could do his experiments on inactivating it, testing it, and so on, up to the point where I remember this very well. In 1955, everybody showed up for their polio vaccination. Enders went to work on the measles vaccine in 1954, and by 1963, his vaccine had become part of the trio, the MMR, measles, mumps, and rubella vaccine that's routinely given to children now to prevent those diseases. So there's a graph of sorts that I'm going to show you next that kind of shows you where we're going to go now from where we've been. And I don't mean it to be over technical, but I think this tells a story. This is a sort of a table or a graph of vaccine innovation. The people who put it together said most vaccines take years to develop, but scientists created multiple vaccines for COVID within a year. And you can see those first three, typhoid fever, meningitis, whooping cough on the left, those are all bacterial diseases. The bacteria were identified as pathogens early on, back in the 1880s, 1890s, and so on. But it took quite a long time before you finally got a vaccine license in the United States. There's polio in the center. Polio was recognized as being caused by something that must have been a virus. That's where the red dot is. Back after 1900, there were attempts to make polio vaccines, but none of them worked. Okay, and in fact, people died while it was being tested. So it's really that period from about 1949 to 1954, where Salk did his work in 55, that we got the first vaccine. But you notice the lines are getting narrower. Mumps is there, that a poor vaccine back in the 1940s, early 1950s. And then there was a new vaccine developed later on, much shorter time. Measles, that was the work by Enders and his group, 9 years. Hepatitis B took a while. The Ebola virus, this virus in the Congo, was identified, the virus was identified, looks like about 1970, but the first vaccine didn't come along until about 2020. And then down at the bottom is this funny little thing called SARS-CoV-2-2. That's the COVID virus. And there's a dot there, and that dot is on 2020, and part of it's red and part of it's blue. And that's what the story is going to be about from here on out. So let's look at the timeline to get a new vaccine when you're dealing with the COVID virus. I remember all this well. I think many of you will too. The first COVID-19 case was reported in the United States around January 20th, 2020, as I recall, out in Washington State, in a rest home. By March 11th, the World Health Organization had declared a pandemic. And by December 28th, 2020, you could line up over in the sterile park right here and wait in line, hoping to get one of the first vaccines. And by February 21st, we were all lined up at our computers, trying desperately to get into the Publix website to get lined up for a shot at one of our local Publix pharmacies. CVS came online, Walgreens, everybody was in the game to register for vaccine shots. And there were three 3 shots. There was the Pfizer, there was the Moderna, and there was the Johnson & Johnson. 3 new vaccines in 1 year, okay? It's a pretty good advance over smallpox, which was 250 years. So what we want to look at is how this happened. How did this come to be? And how on earth did people manage to do this and bring it off in that short a time? What I want to say here is that one of the things you hear about the concerns with the COVID virus was that it took too long to make it. Or it was developed too fast. I'm sorry, it didn't take long enough. Everybody was familiar with the time that went into polio and measles and so forth and so on. And all of a sudden, here's this vaccine that crops up in a year. So what does this mean? How did this happen? All right. So here's the background for COVID vaccine development. The first thing that happened started in the 1960s, the early 1960s, when there were stunning advances in life sciences research generally. The life sciences came into its own with its own tools and its own discoveries. It's equivalent to the impact of quantum theory and physics almost. In particular, the roles of DNA and RNA in cells have been characterized. Everybody knew what DNA did, everybody knew what RNA did. And the science spread from there into looking at all aspects of cell life, cell death, and what have you, in terms of the genetic information that resides in those cells. Techniques were devised for the transfer of DNA or of RNA between cells. They made genetically modified, you got genetically modified organisms. So it was almost routine that you could do almost anything you wanted with DNA or RNA. You just had to find the trick or the technique or the way to do what you wanted to do. And delivery systems were developed to transfer DNA and RNA. There were things like giving cells electrical jolts, voltage jolts. You could force DNA into cells that way, and there were more sophisticated systems as well. 2 delivery systems that we will talk about. One is lipid droplets or liposomes, and I'll show them to you and I'll tell you how they came to be used. And attenuated viruses. These are viruses that have been developed in such a way, their DNA has been modified or their RNA has been modified in such a way that they retain the ability to enter cells, to get into a cell. But once they're in there, they can't do anything. They can't infect the cell. And these become vehicles or delivery systems for DNA and RNA. And overriding over this and overseeing all of it is sort of the central dogma. And I've used this slide is getting pretty old. But basically, it's the whole thing with DNA being the template of the genetic code, RNA being read off of the template, and RNA being used to make proteins. So in the life of a virus, because that's what we've got to talk about, viruses can be DNA viruses or they can be RNA viruses. They can kind of shortcut things. And in either case, that code that's in the RNA or in the DNA, when it's read, can be used to make proteins. In the case of the virus being nasty, those proteins are used to assist in the rebuilding of brand new viruses in the infected cell, kill off the cell, they get out of the cell, and you have a bad cold, you have COVID or whatever. The RNA, by the way, is called messenger RNA or mRNA, and that might ring a bell with you somewhere down the line. You've probably heard that already. They talk about mRNA vaccines, for example. So there are a series of paths that converge in making COVID vaccines, and I'll just take you through these. They're all pretty straightforward. Starting in the period around 1995, a woman named Katalin Karikó, a Hungarian biochemist who was at the University of Pennsylvania, had the idea that if she could figure out how to sneak messenger RNA past the human immune system and into cells, she might be able to do something with it. And I say sneak, she said sneak, because if you just take mRNA and shoot it into your arm, your immune system's sitting there looking and saying, what is that? And you can get immune reactions against foreign messenger RNA. And she spent 2 years trying to do this without very much success until she teamed up and collaborated with a guy named Drew Weissman at the University of Pennsylvania. And they worked out a system where they could use lipid droplets or liposomes to transport messenger RNA into cells. And lo and behold, when they did that, it stimulated the immune system in these cells. So, and a discovery, and it turns out to be an important discovery. They didn't think so at the University of Pennsylvania because at that time, there just wasn't much interest in this particular method. And in fact, Rico was demoted. She couldn't get research money for her interest in this problem. She had kind of a hard time of it, but that's behind her now. She's a senior vice president at BioNTech. You ever heard of them? That's Pfizer. And she and Weissman are also still professors at the University of Pennsylvania. They're very highly regarded professors. They look like this. They're smiling because a lot of people are going around saying they should get the Nobel Prize. The discovery they made way back in the 1990s regarding a way to get RNA into cells and actually stimulate the immune system. So that's part one. And just to show you what liposomes are, this is kind of complicated, but let me tell you how you make liposomes. You take what is essentially fats. Think vegetable oil that'll float on top of a little glass of water. Those blue things up on the big disc, those blue things are the charged parts. They have those charges like water. And those yellow tails are the greasy parts of lipids. You have all kinds of lipids in your membranes. That's what it looks like. And if you just mix the lipids with whatever you want, it could be messenger RNA, in fact. And you give it a really good shaking, the physical forces that are involved there cause the lipids to line up in such a way that they get the water-loving parts on the outside or on the inside and all the greasy stuff in the center. And that looks almost exactly like a cell membrane, which is what's shown down here. And that cell membrane has got the same kind of alignment of lipids. And what happens in the case of liposomes, and there's been a lot of work on this, very, very elegant work that developed this. These liposomes will come along and they'll see a cell membrane and they'll come right up to it. And because of the chemical similarities between the lipids in the liposome and the lipids in the cell membrane, they'll merge. And the liposome will kind of fold into the cell membrane and it will dump those little red things, which I've labeled as messenger RNA. It could be whatever you want to get into the cell. That will be dumped into the interior of the cell, which is down below the screen of your computer at home. So liposome mediated transfer of many different compounds of interest, including messenger RNA, cancer drugs, and what have you, has now been developed using this particular system. Okay, so there's a second path, and I can't show you any pictures of the scientists. These guys are sort of anonymous, these people. But groups at the Johnson & Johnson pharmaceutical company, AstraZeneca in England, Merck here in the United States, all at roughly the same time used a modified non-pathogenic adenovirus to transfer DNA into cells. This is what I talked about earlier. These viruses have been defanged, so to speak. They retain the virus structure that allows them to find a cell and to get into the cell, but once they're there, they've got nothing to do. And they use this technology, the 2000s, I believe, to develop a vaccine against the Ebola virus. You may remember that this outbreak of Ebola. It's a terrible virus. It's a very virulent, very pathogenic. It turned out that the vaccine that was developed, and it might have been the AstraZeneca or the J&J or the Merck, I don't remember which one. 2 shots of this vaccine separated by a month or so conferred strong lasting immunity against Ebola. And it's still, as far as I know, being used where necessary to combat the Ebola virus. So part 3. This is kind of historical. December 2019, the COVID-19 outbreak in Wuhan, China was finally revealed to the world. In about a month, Chinese scientists identified the virus and they published its RNA sequence for the whole world to see. There was no secrecy there. The whole sequence is there, so everybody could look at it. They could see what was in this virus in terms of proteins, the RNA gene sequence, revealed all of that. Later in January, the Center for Disease Control released an artist version of the virus, which you will see shortly, and you will recognize almost everything about it. And February 20th, this is stunning. Okay, so we've got about 2 months gone here. Biologists in the United States and elsewhere, using new techniques, what's called structural biology, were able to determine the structure of this virus's spike protein. And it's the spike protein of the virus that allows it to attack human cells. Okay, so what am I talking about? You've all seen this, I'm sure, on TV or whatever. This is a cartoon. COVID-19 does not have red things on the outside of it and a gray thing as a ball around it and what have you. What those are, the spike protein is there in red. The gray part is a membrane, and inside that membrane is RNA. So COVID is an RNA virus, and the RNA is residing inside the virus. By using electron microscopy and various other techniques, it was easy, it was probably complicated, but it seemed to come very fast. Very easy for structural biologists to see how this virus was actually attacking cells and causing infection. And I have a diagrammatic representation of this. So this is just a cartoon. That sort of dark brown layer is a cell membrane. And if you just move from left to right, the first step in infection is that the COVID virus with its spike protein attaches to these little Y-shaped protrusions on the outside of the membrane. And it attaches very strongly. I think about an interaction with various like tape or whatever. And then what happens is the same sort of thing that happens with liposomes, that there's a fusion between the membranes. The virus particle incorporates into the cell membrane, and that stringy thing coming out is the genetic code of the virus. Okay. So this has been captured by electron microscopy. There are all sorts of pictures about it. You can go in and you can actually find these. They're fairly complicated to look at. But this is the mode of infection. The terrible thing about COVID is that these little Y-shaped protrusions belong to what are called ACE proteins. Angiotensin converting enzyme is an important enzyme in your bloodstream, regulating blood pressure, and it's particularly abundant in areas that have a lot of capillaries, a lot of veins, and what have you, so that the virus, if it gets, say, to your lungs, is really going to have a field day attacking your lungs, and that's why so many people wind up being intubated as part of the recovery process, because the virus really attacks in these areas that have a lot of the ACE protein. Okay, so what everybody figured out from looking at all of this there was science involved, but it basically just deduction. You're going to make a good COVID vaccine. That vaccine has to produce an antibody that blocks the spike protein's ability to bind to human cells, not hymen cells, human cells, spelling. And there were two approaches to this. Approach number one, Pfizer, Moderna. So you sort of know what's coming. All they had to do was go back, compare the spike protein structure from the structures that had already been published and were known with the COVID mRNA, and find in the mRNA where the sequence was that encoded the spike protein of the virus. Number two, synthesize messenger RNA, the sequence, in large amounts. I think that sounds like it might be quite an undertaking, but it turns out that RNA chemistry is very highly developed. You very rapidly synthesize huge amounts of messenger RNA using techniques like the so-called polymerase chain reaction, the PCR test. You've heard of that. It works miraculously to generate large amounts of messenger RNA, very specifically the sequence that you want. And then next, you only have to trap microgram amounts. Microgram is a tiny amount of a gram of messenger RNA in individual liposomes. And the reason for that is that if you can get the mRNA then into the cell, this is a message. It's going to be read hundreds, thousands, millions of times. You're going to generate a lot of protein with a very tiny amount of messenger RNA. The way they do this is carefully shielded by patents. But I saw a Pfizer scientist on television being interviewed about this, and he sort of led on how they were doing it. And it's just basically that you mix the RNA with the lipids, and you swirl everything up into a mass. And what comes out of that are the liposomes, and has mRNA trapped in the liposomes. And now you have your vaccine. You vaccinate. If you've got a Pfizer or you've got a Moderna, you were vaccinated with liposomes. And those liposomes, the lipids that are in those liposomes are trained to go for cell membranes. So those liposomes will get into cells, carrying the messenger RNA with them. Your cells are just sitting there waiting for something to do, and the cells can read messenger RNA, and they read it, and they synthesize the spike protein. And once the spike protein is seen by your cells, antibodies are produced, and you're vaccinated. And so the only other thing that had to be done was to make the antibody of the spike protein in such a way that it'll neutralize the virus binding, and we know that that works. Okay, so that's the first way that it was done. The second way is the J&J and the AstraZeneca approach. And that's when, remember that these pharmaceutical companies had specialized in using adenoviruses, viruses that were trained to infect cells, but not to do anything once they got into cells. So they just synthesized the spike protein DNA sequence, because the DNA sequence can be read to make the RNA, which is what in the virus, and then insert that DNA into the adenovirus. Okay. And then after that, you get vaccinated with the adenovirus that can't do anything to you except get into your cells and release the DNA that codes for the spike protein, and the cells produce antibody that binds to the spike protein. So two rather different approaches, but both giving the same result, that you get the message for the spike protein into the cells, and the cells then produce the spike protein, and your cells produce the antibody that's needed to fight off the virus. So there's some add-ons here. For example, you had to do additional testing of all these vaccines. The first that I knew of was the Pfizer vaccine, which was vaccinated shot into volunteers. There were about 40,000 to start, and it's reached millions now among all the producers. And you're looking for any sign of a problem with having the spike protein in the recipient of a shot. You cannot get the COVID disease from the spike protein. The spike protein could do nothing but initiate production of antibodies, which is the vaccine. Then you run and apply for emergency use authorization, the EUA from the FDA, and everybody got that. And because of the technology that's used here, it's very easy to produce millions of vials of vaccine and fairly quickly and very short periods of time. And you can have either have an mRNA virus, an mRNA vaccine, or you can use the DNA virus and you can get to it that way. Okay, so results. as of a couple of days ago anyway, 574 million vaccine shots have been administered in the United States, 213,000 people vaccinated, 65% of the population fully vaccinated. Very few incidents of any kind. However, there was an issue with the J&J vaccine. There were blood clots in women of a certain age, and there were 9 deaths and 17 million vaccinations. It works out to be a tiny percent, but it is something that you have to be aware of. You have to be careful about, and it can be offset. These people can be identified, and they can be given another vaccine. As far as is known, blood clotting is not a problem with the other vaccines, the Moderna and the Pfizer. The other thing that's come up, and you've probably all heard of this, the Pfizer and the Moderna vaccinations wane. That means they become less effective over time. And it seems to be about 6 months. Most vaccines will do this. I mean, you get a shot and your B cells go to work and you get antibody and everything. But after a certain period of time, they begin to run down or wear out. They may be provoked and come back again, but your antibody levels are down. J&J is more durable. It's still being tested, and it seems probably because it's using DNA, perhaps, which is more stable than RNA, is able to generate antibodies for a longer period of time. And we've seen, as you know, probably 5 to 10% breakthrough infections. So booster shots have been recommended. Many people have had their booster shots, which means that, generally speaking, against the original virus and against the Delta variant and against the Omicron variant, these vaccines are pretty effective, 90 to 95% effective overall, which is very good. So let's talk about the variants, the mutations in COVID RNA. There's a reason for this. The original COVID virus is just like any other virus. These viruses are not very tidy when it comes to copying their genetic codes. In other words, when they've infected a cell and they're making new viruses, let's put it this way, in humans, our DNA copying exists with extremely high fidelity, with errors that can run maybe to as little as maybe 1 in a billion bases in DNA, maybe a few more. But our enzymes that do this, they have an error correction facility, and they'll be checking what they're doing. And so mutants don't sneak through in animal cells like they do in bacteria sometimes, and certainly how they do in viruses. So the errors that you get are the ones that produce these variants or these mutations, and that would be like Delta and Omicron. And the issue here is that, and we'll get to this in a pictorial way in just a second, mutations like these in the virus, spike protein, can result in changes in the amino acids or in the removal of an amino acid. And if that happens, these changes can affect the extent to which the antibodies from vaccination with the original spike protein can still recognize the new spike protein, all right? Now, the answer seems to be that we get pretty good, maybe excellent protection from the original, the vaccine against the original spike protein. It doesn't seem that the Omicron spike protein is getting past the vaccine in any large amounts, and certainly Delta didn't either. So what do these look like? This is as scientific as we're going to get. I don't mean to scare you with this, but these are the structures of the spike proteins from Delta and Omicron. Don't have to worry about the spirally stuff and everything else. These are the strands of amino acids. It's the colors that are here. So the original virus protein, spike protein, isn't present. It wouldn't have any of these colors. The Delta, these colors, and it says they're white ones here. I've never been able to really find them well. But the colors in red indicate where an amino acid has been removed. It just isn't there anymore compared to the original spike protein and the original COVID virus. The blue dots represent places where a new amino acid has been switched in place of an old amino acid. So in the Delta, I don't know, I count maybe 18 at most, maybe fewer than 18 places where there have been these changes. And so it's not something that's really, really drastic. It may explain why Delta was not really very good at slipping past the vaccines. But look at Omicron. This virus has got huge numbers of amino acid changes and more deletions than in Delta. So this spike protein causes the virus to be more infective, but the lore has it from now having looked at all the data that the infection is not as serious or as severe as Delta or the original, okay? And again, the issue is that when you change these amino acids, you may be changing or maybe affecting the ability of our antibodies against the original to deal with an Omicron protein that's sticking on a virus. So life with the variants, the mutations. Okay, so I already said you can affect antibody binding, and the Omicron variation mutations, you've seen those now. The variant, for some reason, is more infective and partially resistant to antibody protection, but it doesn't seem to be as deadly as the original or as the Delta was. A problem that comes up when I hear the epidemiologists and the immunologists talk about this is that more variants may appear in the future. There is that possibility, and this comes about in the following way, that if you have in a population, we're about 65% totally vaccinated in the United States. You have a population of about 35% unvaccinated people, where viral infections will result in the virus replicating itself. Those viruses may generate future mutations. The same is true around the world, where you have low rates of vaccination may result in higher rates of mutation, and some of those mutations may be problems. And Pfizer is already said to be in the process of setting up for producing a new vaccine that's directed specifically against Omicron. I don't know for sure that they're actually going to go forward with this. The last I heard that they kind of put this on the back burner, and it was not decided that there was going to be a need for it because of the original vaccine doing such a good job against Omicron. But the point is that for anything coming down the road, any pandemic, any viral infection or whatever, where you can get the information that was available for COVID, it's possible for pharmaceutical companies to very rapidly, very quickly produce a new vaccine using the trick of picking the surface receptors, the surface binding proteins, and going after them with the antibodies that you can produce with the right immunizations. So conclusions, human immune system is a wonderful thing. It's flexible, it gives great defense against disease. We can be manipulated by vaccination in such a way that we can resist things that just pop up on the map all of a sudden. The polio is gone. Measles isn't suppressed completely, but measles is pretty low, and COVID is on the run. Remember that from what I've told you, that the vaccine developments in the 20th century needed many experiments and testing of viruses treated to lower their infectivity. And that's where people have the impression that for polio, for example, and for measles, it took a long time to develop the vaccine. But also remember that what we've been talking about, that in the 20th century, molecular biology progress and biochemistry made rapid progress in areas that benefited vaccine development. And in 2020, 25 years of research. So it was a long time. It was just there and nobody knew we were going to need it. 25 years of research results coalesced to produce effective COVID vaccines in less than 1 year. So that's all I have to say, except that I would be very happy to take any questions that you might have. Hmm. Jordan seems to have disappeared. I don't know how to open you up, Ken. There, Jordan, we have a question.
Oh, sorry, I was using the ladies' room. Sorry. Yeah, we do have one question, so let me allow Mr. Selger to talk.
This microphone's off.
Yeah, Mr. Selger. Okay, there we go.
Gotcha.
Great, thank you. Very good talk, Charlie.
Thank you, Ken.
One of the things I remember hearing someplace was that Moderna got some DARPA money early on before the virus started to develop vaccines rapidly. Do you know if that's true? Have you heard the same thing?
I heard the same thing, yeah. And there was actually an interview on 60 Minutes before all of this happened with one of their senior researchers, and they were talking about this. They were talking about being able to find a way to get a virus's genetic code into cells and use it to make antibodies. So, but they were all kind of flying blind at that point. And the money, Moderna got government money, and Pfizer went on their own. They used their own funding to develop the vaccines. J&J is a company, they funded their own as well, I think. But yeah, I saw this thinking, I'd heard about the virus and I'd heard about COVID and everything, but I thought that would be a really clever way of going about it. And it was.
Okay, thank you.
Yeah. Mr.
All right,
McDowell.
we have two more questions too. There, if you look at the bottom, there's a Q&A that popped up. And then Mr. McDowell.
The chicken pox is a virus, and it can, even after it's over when you're a child, it can remain in your body and apparently cause shingles. Is that a unique to chickenpox virus, or can something else can that be similar to what we see with COVID, or what we could see with COVID?
Okay, I will give this my best shot, but there's an MD lurking in the audience, and if I go astray, I would expect Dr. Colfazer to correct me. There are viruses that are called retroviruses, and they're particularly nasty because what they do is they infect and then they just plant their nucleic acid in your nucleic acid, okay, and hide it. And at a later time, okay, it can be activated, and stress, various signals of various kinds can activate the signal, and you get the virus, okay. And HIV is a retrovirus, and I think that maybe the chickenpox shingles thing is a retrovirus as well, but I can't be absolutely certain about that.
All right, one more question here. I think Mrs. Yoakum, I think you're.
No, it's Peter Colfazer. It's got his microphone off.
Okay.
Over there. Okay, can you hear me?
Yeah, Peter.
Yeah, I mean, as a radiologist, I'm not an expert on infectious disease, but I suspect that you have other viruses like the polio virus that many years later people have syndromes secondary to having had polio at a young age, and possibly some of this delayed COVID that we're hearing about, probably or possibly, I'm only speculating, could represent some kind of persistence of the COVID virus in some dormant form or partially active form, which would be similar to the shingles and the chickenpox thing. But again, that's really not my area of expertise. About all I have to say.
Thank you. So Mrs. Yoakum did have a question. She typed it. She said, why does the polio vaccine not wane, or does it wane? I don't know if she meant work.
Why doesn't it wane? I think that the answer to that is, I don't know. I think ultimately with polio vaccinations, you got more than one shot. And I think it was the number of shots you basically got long lasting or permanent immunity. Your B cells were really tuned up to go after it. But I can't answer that better than that.
All right. Was there any other questions?
I'd like to ask one more.
Really quick, I am going to open up that poll. So just in the background, as you're listening to some of these question and answers, if you could take that survey for me. Thank you.
Initially, when the COVID-19 was apparent, there was discussion that it began in Wuhan, and of course the lab there was suspect. But others said that one of the reasons it began there is because Wuhan has a large number of wet markets where they sell meat, and most of the meat, a lot of the meat, is from what we would call exotic animals, and that's where it began. And shortly after it was detected, the Chinese closed down wet markets, particularly in Wuhan, but in other locations in China. But they did not stop the exportation of this meat. And so the theory was that that's how it spread. Have you heard anything about that, Charlie, or anyone?
I'm familiar with the accusation that it might have escaped from the lab, but there have been all of these investigations, World Health Organization, people in the United States, that were inconclusive. Certainly it could have spread. Well, that lab, I think, dealt with bats and bat viruses. Yes. And that's where, that's where it could have come from, either indirectly or directly or whatever. But from what I've read, and the last time I looked was probably a month ago, nobody's willing to come out and say definitively it escaped from the lab, it was in the wet markets. The chances are good of any of those being the origin of it. It's an animal virus, and it's a nasty one.
One more question, Charlie, if you don't mind.
Sure.
Sanofi Pasteur is a significant researcher and producer of vaccines, particularly the flu vaccine annually. Why weren't they in the forefront of this, or is COVID significantly different from what we know as annual flu?
Well, the little I know about flu vaccine production is that it's a completely different process. Totally different. And I think what they do, if I'm not mistaken, is that they try and look at a crystal ball and decide what sort of mix they have to have of viruses to put into the flu vaccine for a given year. And what you're getting when you get shot is you're getting a mixture based on their best guess of what's going to be in the environment. They're not using this. They're not, as far as I know, they're not using this DNA/RNA technology. I think lots of chicken eggs go into making flu vaccine. And I think that's one of the problems with flu vaccine. If you're allergic to chicken yolk, that might be a problem. Although I don't think people are affected by that very much. But the other thing, of course, is that this whole thing was pretty much a U.S. operation, except for AstraZeneca in England, which had been collaborating. Well, J&J is also European. The headquarters are in Belgium. So they're on a different track experimentally in how they do this. The best I can say about flu vaccine versus COVID vaccine.
Thank you.
All
So-
right, any more questions here?
To return to Wuhan just a minute, the theory was that it was pangolin, the meat of the pangolin, which was the genesis of the original COVID-19. And that's why they cleared so many of the wild animal wet markets, they call them in China, and it grew from there. You never heard anything about that other than the poor little pangolin being.
No, I know I've seen pictures of these poor little animals, but there was just one of many the theories about where the virus could have come from.
I'd like to comment on that. I recently did some research, and it turns out that the coronavirus is 96% identical to known existing bat viruses. So it's very, very close, and it doesn't take much of a change to leap into the current version that we have. As far as the lab doing it, the question is, did the lab have some and accidentally let it loose? It's a possibility, but most researchers believe it's highly unlikely. One researcher did some interesting research where he plotted where the cases showed up in the early cases in actually November and December, and they all were around the Wuhan wet market. The
Right.
lab is located about 10 miles from there, and none of the cases that were showed up were anywhere near the Wuhan lab. So all the cases turned out to be clustered around the markets, making a pretty strong argument that it was from the market that the virus originated.
Yeah, I saw that as well. I think there was also another study that had a hot spot or something, some distance out of Wuhan, more than 10 miles from the lab, where there was a cluster of infections. So we'll never know, probably.
It's somewhat suspicious how the Chinese were started in the WHO and allowing them in and making certain that the WHO was staffed by people who either wouldn't accuse China or were not able to really analyze the problem. And that was in the New York Times and the Wall Street Journal. I don't know how true that is, but it certainly took a hell of a long time for the WHO to get their acts together and get over there. And they were limited in what they could do when they were there. Now, is that true or is that just suspicion?
That isn't science, and so I can't comment on it.
Well, now, politics is science. You just...
Oh, come on. If politics is science, cows can fly.
I'm sorry, I don't want to even go there.
Okay.
Well, thank you, Mr. Yoakum. Another awesome class. Just a friendly reminder that we actually are switching back to in-person sound learning classes come March. Tons of great options for you to choose from. So if you want to take a look at the upcoming events and sign up now, they're all open. I believe Mr. Yoakum is actually teaching another one at the end of the month. They will all be outdoors behind the admin building on the patio. social distancing outdoors. So we're still being cautious, but we wanted to switch back to in person because we feel like those are, more interactive classes. So there, like I said, tons of great options to choose from if you want to look under the upcoming events. But for now, we still have all of our virtual classes that will be up on YouTube. I will make sure to get this one uploaded for everyone tomorrow if you want to take a look back at any of the classes.
So Jordan, I have a question for you.
Mhm.
Have we actually tried running the projector in that tennis patio area? Do we have sufficiently dark enough so that we can see it?
Travis, I know that Travis has done a few meetings out there with the projector, and he said it's worked pretty well. I haven't personally seen it myself, but like I said, Travis has used it for a handful of meetings.
Okay.
We do have our first class actually Tuesday with the Shy Wolf Sanctuary, so we'll make them test it out. But it should be good because he said that he's used it a few times for meetings. So
Okay.
I think
Very
it'll be a
good.
good alternative. Yeah.
And there are no pendula close to that outdoor patio, are there?
I'm sorry, ask that again.
There are no pendula close to that outdoor patio. So we're safe.
Yes, correct.
Yes, right. No Chinese animals.
Yeah. All right. Well, thank you, everyone.
Okay.
Thank you. Well done,
Thank you.
Charlie.
Mhm.
Thank you.
Bye.